Hello everyone, welcome to Pearson Bio Academy. If you are new to my channel, this is Fazna Imran and this is the place where you can top up your biology knowledge with our past paper explanation through very short and clear video series. Let's get diving. Today we'll be focusing on Pearson Edexcel International Advanced Level Paper Reference WBI 1101 Unit 1 Molecules, Diet, Transport and Health. So this is the course outline. If you have watched my all video series, then you can recognize. So this video is going to cover the chemistry of life. And under chemistry of life, we have carbohydrates, chemistry of water, proteins, lipids, and enzymes. And today we will be tackling questions on proteins. Welcome to part 2 of the protein video series. If you haven't watched part 1 yet, I encourage you to check it out first. In that video, we have explored about amino acids and tackled questions related to them. We know that proteins are made up of many number of amino acids linked together by peptide bonds. Here I'm going to show you how a peptide bond is formed between two amino acids. First, it's important to note that this is a condensation reaction. So during this process, one water molecule is removed, which is why it's called condensation. As a result, the peptide bond will be formed. This image is taken directly from your student book, which is why I have included it here. And you now understand how peptide bonds form from the early animation, but it's essential to know how to illustrate them in a diagram. In this example, you can see how amino acids join with the removal of an H2O molecule in a condensation reaction. On the flip side, this is a reversible process. If you add one water molecule back to the bond, the dipeptide can split into two amino acids. And when two amino acids are joined together, it's called a dipeptide. Now let's quickly go through some questions about peptide bonds and protein formation. And the first question is from the October 2020 paper. You are provided with a short paragraph and you are asked to complete it by adding the most appropriate word or words on the dotted lines. And here is the paragraph. The primary structure of a protein is the specific sequence of amino acids joined together by peptide bonds. And these bonds are formed between the amino group of one amino acids and the carboxyl group of an adjacent amino acids by a condensation reaction. And these bonds are formed during the stage of protein synthesis called translation. The next question is from the June 2021 paper. In this question, you are given a table that includes all polymers, but I have specifically picked out this section on proteins. And the table asks you to identify the monomers, the elements present in each monomer, and the type of bond between the monomers. And for proteins, your task is to complete the table by listing the elements present in an amino acid. And you know these elements are carbon, hydrogen, oxygen, sulfur, and nitrogen. However, you can omit sulfur since it's only present in the amino acid called cysteine. The next question is from the October 2021 paper. In this question, you are provided with a table and asked to identify the bond between two amino acids and the type of reaction. Thus, answer is so simple. You know the bond is the peptide bond and the reaction is a condensation reaction. Interestingly, the exact same question appeared again in the May 2024 paper. So the answer is same. And this is a great example of why studying past papers effectively is so important. Now let's see the structures of protein. They are primary structure, secondary structure, tertiary structure, and finally its quaternary structure. First, we will look about the primary structure. So these different color balls are representing different amino acids. So they will join together and form an amino acid chain. And if you look the bonds between two amino acids, it's just peptide bond only. Next, secondary structure. The primary structures of a protein falls into specific secondary structures such as alpha helices and beta sheets. Alpha helices form due to hydrogen bonds between the amino and carboxyl groups within the same polypeptide chain. The beta sheets, on the other hand, arise from hydrogen bonding between different segments of the chain, forming a pleated sheet-like structure. The tertiary structure of a protein refers to its raw 3D shape formed by the folding and interactions of secondary structure elements like alpha helices and beta sheets. And this structure is stabilized by various bonds including hydrogen bonds, disulfide bonds, and ionic bonds between R groups of amino acids or side chains. In the quaternary structure, two or more three-dimensional polypeptide chains join together to form a single functional protein complex. And a classic example is hemoglobin, which consists of four subunits working together to transport oxygen efficiently. Let's quickly analyze this question which appears in 2020 October paper. This question is mainly about the bonds which occur in different structural protein. The first one, secondary structure. If you can remember, the secondary structures are formed due to the hydrogen bond. Therefore, hydrogen bonds only within secondary structure. And if you take three-dimensional structure, there are various interactions or bonds are present such as hydrogen bond, disulfide bond, and ionic bond. So for this table, the both hydrogen and ionic bonds are present within three-dimensional structure. So this question is from 2023 May paper and it's focused on the correct statements for proteins. The first statement states protein contain only the elements carbon, hydrogen and nitrogen. You know that it contains oxygen also. The second statement, proteins can be made of more than one polypeptide chain. I can here give you a good example. It's hemoglobin. You can see it's made up of four subunits, or four polypeptide chain. Therefore, the second statement is correct. And the third one, proteins contain hydrogen bond. So previously I explained you the both secondary structure, tertiary and quaternary structures contain hydrogen bonds. Therefore, the third statement is correct. So for this question, two statements are correct and your final answer is C. The diagram here is illustrated and you can see there is an A chain and a B chain linked by a connecting region. And the question is here, the connecting region is removed from proinsulin, leaving the A chain connected to the B chain by disulfide bridges. Which row of the table shows the position of an amino group and a carboxyl group? We know that in the chain, amino acids are connected by peptide bonds which form between the amino group of one amino acid and the carboxyl group of the adjacent amino group. So in the given diagram, it's stated that one end of the B chain contains an amino group. So the other end must contain a carboxyl group. The connecting region joining with the B chain should then start with an amino group. Similarly, the other end of the connecting region should contain a carboxyl group. One end of the A chain should also have an amino group at position R, while the other opposite end should contain a carboxyl group. I hope you now understand the concept. In the given position, there are carboxyl groups at positions P and Q and only an amino group at position R. For the table, position R should be listed under the amino group while either position P or Q can be listed under the carboxyl group. Therefore, the most suitable answer here is A. And you can simply eliminate the other options because the amino group is only present at position R. The next part is a small calculation. The question states that proinsulin is composed of 84 amino acids and insulin is composed of 51 amino acids. And you have to find out how much percentage of proinsulin is the connecting region. So we will do this question step by step. So very first you should know the total number of amino acids. Those are in the proinsulin. It's 84. Now you have to find out the number of amino acids in the connecting region. For this you have to subtract the 51 from 84. It's 33. Then we have to find out the percentage. So that you can use this simple formula. Number of amino acids in the connecting region over total number of amino acids in the proinsulin times 100 because you have to present your answer in percentage. And the final calculation 33 over 84 and you have to take it for 100 times. It's simply 39.29%. The next question also a simple calculation and the question states there are 3 disulfide bridges in insulin. Each disulfide bridge is formed only between the R groups of 2 cysteine amino acids. And you have to find out the ratio of amino acids that forms disulfide bridges to those not forming disulfide bridges in insulin. Let's tackle this question step by step. So this is the diagram. If you look at the diagram carefully, then you will notice there are 3 disulfide bridges. It means 6 amino acids take involved in forming these disulfide bridges. And now the amino acids forming disulfide bridges is 6 and the number of amino acids that not forming the disulfide bridges is 51 minus 6. It's 45. So the ratio is 6:45. If you simplify it and the final ratio is 0.13:1. And the next part of the same question is suggest how insulin is synthesized from proinsulin. You can refer to the diagram to help frame your answers. The answer is quite simple. At position P, and R, a hydrolysis reaction occurs to separate the connecting region. If you recall, during a hydrolysis reaction, one water molecule will be added to the peptide bond, which causing the bonded amino acids to split apart. Please keep remember, all these hydrolysis reactions are taken place under enzyme control. Now let's tackle the keywords. The keywords are hydrolysis, enzymes, peptide bonds broken. And you can write your answer like this. By using enzymatic hydrolysis reaction that break the peptide bonds between P-N connecting region and R-N connecting region, insulin is being synthesized. The next question is from 2024 January paper. Actually, this question should come under the characteristics of R groups of amino acids. However, I have brought up here. And the question states channel proteins are involved in facilitated diffusion. Which row of the table describes the charge on the R groups on the amino acids of a channel protein? You have to look the charge on R groups facing inside the channel and the facing the rest of the membrane. This picture illustrates a cell membrane structure with a nice channel protein. If you look at the diagram carefully, you can see that the outside of this channel protein are facing to fatty acid, which is nonpolar. Therefore, they should contain R nonpolar groups. And in the other side, the inside the channel protein or through the pore, they have to transport polar molecules. Therefore, definitely they should contain R polar groups. So now answer is very straightforward. Charge on R groups facing inside the channel are mostly polar, and charge on R groups facing the rest of the membrane are mostly nonpolar. Therefore, the best answer is here B. Let's quickly analyze this question. Describe how a chain of amino acids forms an enzyme molecule. First, the amino acid chains represents the primary structure, and this converts into the secondary structure, which can be an alpha helix or beta sheet, and this stabilized by hydrogen bonds between amino acids. Next, the tertiary structure forms as the chain falls into a 3D shape, stabilized by hydrogen disulfide and ionic bonds between R groups. Finally, the enzyme is completed with hydrophilic groups on the outside and active sites ready for function. Now, let's tackle the answer using the keywords. First, the amino acid chain forms a secondary structure into an alpha helix or beta pleated sheet stabilized by hydrogen bond between the amino acids. The secondary structure then falls further into a 3D or tertiary structure where different types of bonds including hydrogen bond, disulfide bond and ionic bonds form between R groups. And this folding results in a globular structure with an active site where hydrophilic R groups are arranged on the outer surface. The next question is from 2021 January paper. This question is about the hemoglobin structure, and you know the P here label is the heme group, and the question state the role of the structure label P, and you know it binds to an oxygen molecule. Next question is from the January 2021 paper. Here you are given with the structures of adult hemoglobin and fetal hemoglobin. If you look at the diagram carefully, you can see that adult hemoglobin contains 2 alpha chains and 2 beta chains, whereas fetal hemoglobin contains 2 alpha chains and 2 gamma chains. Therefore, the structural difference between them is the presence of 2 beta chains in adult hemoglobin and 2 gamma chains in fetal hemoglobin. So here again you are given with a table which shows the number of amino acids in each type of chain. In alpha they have 141 amino acid, and in beta they contain 146, and gamma again it 146. So the amino acids in the alpha chains are the same in adult and fetal hemoglobin. The beta and gamma chains differ in 39 of their amino acids. So 146 minus 39, 109 amino acids are common in both beta and gamma chains. And you have to calculate the percentage of amino acids that are different in adult and fetal hemoglobin. Now we will do this calculation step by step. First, we have to find out the number of total amino acids in hemoglobin. Amino acids in 2 alpha chains, it's 2 times 141 and it's 282. And amino acids in 2 beta or 2 gamma chains. For each beta chain contains, it's 146, so 2 times 146, it's 292. So altogether, it contains 574 amino acids. Now we will find out the number of different amino acids. For each chain, it differs by 39 and it contains 2 chains. Therefore, 2 times 39, it's 78. Now we have to calculate the percentage of amino acids that are different. You can use this formula. Number of different amino acids over total number of amino acid times 100, because we have to take the percentage. And final formula, 78 divided by 574 times 100, and it's equals to 30.5%. So the adult hemoglobin differs by 13.5% from fetal hemoglobin. Here is a simple model of hemoglobin molecule, which is a quaternary structure, and it consists of 2 alpha subunits and 2 beta subunits. Furthermore, they contain heme group in each subunit, which is binds to the oxygen molecule. Now let's write the answer using keywords. Keywords are consist of 4 subunits, alpha chains and beta chains, heme group, oxygen binding site. Hemoglobin is a quaternary protein composed of 4 subunits. They are 2 alpha chains and 2 beta chains. Each subunit contains a heme group. The heme group consists of an iron at its center, which is responsible for binding oxygen. I hope this video was clear and informative. If you haven't already subscribed to Pearson Bio Academy, please subscribe to stay updated with all our future content. Don't forget to hit the like button if you found this helpful. Thank you for watching and stay tuned for more videos coming soon.